The future of genomics, and in particular gene sequencing, is very promising. It's a multi-billion-dollar market, and it's growing. It's estimated that within a few years, we'll have sub-$1,000 genome sequences. But as Dr. Valley at Johns Hopkins has said, we have the $1,000 genome, but it still requires the $1 million interpretation. We're positioning Cytognomix to be one of the companies involved in the interpretation of genomes. I'm here with John Mukaki, one of the developers of software that we're about to release as a new product for genome analysis. We're here in our laboratories, standing in front of an instrument that we use to analyze breast cancer genes. We use this software to actually interpret gene variants and mutations that are found in women with inherited breast cancer. And John was one of the principals who helped us discover many of these new mutations. So I'm here with Ben Shirley, who's one of the developers of the software that we're about to release. So we're going to do a worldwide release of this software, which is used by genome analysis laboratories to analyze variants in the human genome sequence. This is now a fairly large business, and there's lots and lots of sequence being produced all over the world in many academic, pharmaceutical, and biotechnology companies. And so Ben has developed some very fast software that allows us to look at hundreds of thousands of variants in a matter of a few hours, and to determine which of those gene variants could be responsible for disease. This is one of the machines that we see in many of these genomics laboratories. These machines produce billions of nucleotides of sequence information in a matter of a week or a few days. And we solve the problem of being able to sift through that very large number of variants to try to identify those which are important for disease predisposition or which cause diseases. Our work actually involves cancer genetics, but it's actually a product that can be used for interpretation of genome sequences, of any genome sequence. So our plan, once this product is released, is to develop variants of the software that analyze other types of mutations in the human genome, because our technology is patented. We are also planning to produce this software to analyze other types of genomes, for example, in experimental animals and in plants. And we hope that this will allow cytogenomics to become the leader in this particular marketplace.